My name is Carreras González. I am a postdoc in an analytical development department in a company called Bio-Techne. Bio-Techne is a CDMO specialized in the manufacture of gene therapy products, specialized in the treatment of rare diseases such as juvenile Parkinson or SPG50. So our mission is to support our clients from the developmental stage of the projects to the commercial manufacture of the gene therapies, ensuring that the high quality standards are met under the GMP conditions. I work in the analytical development department, so my job consists of developing new analytical techniques to characterize our viral vector products and then to ensure that they meet the GMP standards. It's important to mention that for analytical development under GMP conditions, this method has to be really robust, highly reproducible, and meet several requirements in terms of linearity, accuracy, precision. So it takes some time to do this job. So my job right now is focused on the Maurice instrument, which is a really versatile and user-friendly equipment. And currently we use CE-SDS, which is capillary electrophoresis, to perform the purity assessment of our drug products. And we have also developed a method to do a deep study into the impurities that might appear in our drug substance to determine the nature, if they belong to protein, DNA, because CE-SDS can detect either proteins or DNA. But now we have started developing CE-IEF to monitor the stability of our samples along the time and to have also a deep understanding on the post-translational modification of our samples. And also we would like to develop it to assess the identity of our samples, because AAVs can be of several serotypes from 1 to 10. Each of them has a different characteristics and different target tissues, so it's really handling. It's really convenient for gene therapy manufacturing, but we need to respond to the identity question regarding our sample. And right now it is answered by Western blot, but it cannot distinguish between the serotypes of AAV. But now we would like to develop CE-IEF to answer this identity question and to unequivocally determine the serotype of our sample.